Binding of the T-cell receptor complex directs the release of effector molecules and focuses them on the target cell. When binding to peptide MHC complexes, the T-cell receptor molecules and their cross-linked co-receptors cluster at the site of cell-to-cell contact. Clustering of the T-cell receptors then signals a reorientation of the cytoskeleton that polarizes the effector cell so as to focus the release of effector molecules at the site of contact with the target cell. Polarization of the cell starts with the local reorganization of the cortical actin cytoskeleton at the site of contact. This in turn leads to the reorientation of the microtubule organizing center, MTOC, the center from which the microtubule cytoskeleton is produced, and of the Golgi apparatus, GA, through which most proteins destined for secretion travel. In the cytotoxic T cell, the cytoskeletal reorientation focuses exocytosis of the preformed lytic granules at the site of contact with its target cell. The polarization of a T cell also focuses the secretion of soluble effector molecules whose synthesis is induced de novo by ligation of the T cell receptor. For example, the secreted cytokine interleukin-4, which is the principal effector molecule of Th2 cells, is confined and concentrated at the site of contact with the target cell. It has been shown that the enhanced binding of LFA1 to ICAM1 creates a molecular seal surrounding the clustered T cell receptors, CD4 co-receptors, and CD28 molecules. Thus, the antigen-specific T cell receptor controls the delivery of effector signals in three ways. It induces a stable binding of effector cells to their specific target cells to create a tightly held, narrow space in which effector molecules can be concentrated. It focuses their delivery at the site of contact by inducing a reorientation of the secretory apparatus of the effector cell, and it triggers their synthesis and or release. All these receptor-coordinated mechanisms contribute to the selective action of effector molecules on the target cell bearing specific antigen. In this way, effector T cell activity is highly selective for those target cells that display antigen, although the effector molecules themselves are not antigen-specific. And that's binding of the T cell receptor complex directs the release of effector molecules and focuses them on the target cell. Have a great day.